Cholesterol is inside every single cell of your body right now. And without it, every one of those cells would collapse within seconds. Every membrane of every one of your 37 trillion cells is held together by cholesterol. It is the structural glue of your entire body. Cholesterol is also the raw material your body uses to make every single steroid hormone: testosterone, estrogen, cortisol, aldosterone, and vitamin D and bile acids. And yet this same molecule, essential everywhere, constantly made, is blamed for killing more people than almost any other substance on Earth. The truth about cholesterol is far more complicated and far more interesting than anything you have been told. And it starts with understanding what cholesterol actually is at the molecular level. Cholesterol is a lipid, a fat-soluble molecule, and its structure is like nothing else in biology. That rigid flat ring structure is what makes cholesterol so uniquely useful and so uniquely dangerous. Your body makes about 800 milligrams of cholesterol every single day, mostly in your liver, because it is so critically important that your body cannot rely on getting it from food alone. The key regulatory step in cholesterol production is controlled by an enzyme in the liver. And this is exactly the step that statin drugs target to reduce cholesterol synthesis. The cell membrane is not a simple bag. It is a precisely engineered double layer of molecules, and cholesterol is woven into it at every point. Cholesterol controls how fluid, or how rigid the membrane is, a property called membrane fluidity. And getting this balance exactly right is critical for cell survival. At body temperature, without cholesterol, the membrane would be too fluid and leaky. At cold temperatures without cholesterol, it would freeze solid and shatter. Cholesterol also clusters with specific proteins in the membrane to form organized platforms called lipid rafts, specialized regions where certain cell signaling reactions happen. Every steroid hormone in your body, every single one, is made directly from cholesterol by removing atoms and adding functional groups to the four-ring structure. In your adrenal gland, cholesterol is converted to cortisol, your stress hormone, through a chain of reactions occurring partly in the mitochondria and partly in the internal membrane network. In the testes and ovaries, the same cholesterol molecule is the starting point for testosterone and estrogen, which is why chronic stress that depletes cholesterol availability can impair reproductive hormone production. Cholesterol cannot dissolve in blood. Blood is water-based, and cholesterol is fat-soluble. So the body had to invent a remarkable solution to the transport problem. These transport vehicles are called lipoproteins, particles made of lipids and proteins. And different types of lipoproteins carry cholesterol in different directions to different destinations. The journey begins in the intestine after you eat fat. Large particles are assembled in the intestinal wall cells, loaded with dietary fat and cholesterol, and sent into the lymphatic system before entering the blood. The liver is the central hub of cholesterol transport. It makes lipoprotein particles, loads them with cholesterol and fat, and sends them out into the bloodstream to supply every tissue in the body. As these liver-made particles circulate in the blood, they deliver fat to muscle and fat tissue. And as the fat is removed, the particle shrinks and transforms, becoming progressively denser and smaller. These small, dense particles are now almost pure cholesterol, and their job is to return to the liver to have that cholesterol recycled or disposed of. The number of these receptors on liver cells directly determines how much cholesterol is cleared from the blood. Fewer receptors means more cholesterol stays in circulation longer. The process that leads to heart attack starts not in the heart, but in the wall of an artery, decades before any symptom appears. The first event is damage to the endothelium, the single cell layer lining the artery, caused by turbulent blood flow, high blood pressure, cigarette smoke, or chronically elevated blood sugar. The damaged endothelial cells change their surface. They begin displaying proteins that attract circulating immune cells, and they become leaky, allowing lipoprotein particles to enter the artery wall. Once inside the artery wall, these particles are chemically modified, oxidized, by reactive molecules already present in the tissue. And this oxidized form is what triggers the true inflammatory disaster. The immune cells that entered the artery wall now engulf these oxidized particles, but they were not designed to handle this load, and they become trapped and engorged, turning into what pathologists call foam cells. These foam cells die inside the artery wall, releasing their fat contents, which forms a soft, lipid-rich core inside the growing plaque. Smooth muscle cells migrate to the site and produce collagen, forming a fibrous cap over the lipid core. This cap is what separates the dangerous lipid pool from the blood. A stable plaque with a thick, fibrous cap may narrow the artery for decades without causing a heart attack. But a vulnerable plaque with a thin cap and a large lipid core can rupture without warning. When a vulnerable plaque ruptures, the lipid core is exposed to the blood, and the blood reacts immediately with a massive clot-forming response that can completely block the artery in minutes. Not all cholesterol transport leads to plaque. The body also has a sophisticated reverse system that collects excess cholesterol from tissues and artery walls and returns it to the liver for disposal. The protein on this reverse transport particle acts like a vacuum cleaner for cholesterol, pulling it out of arterial plaques, foam cells, and peripheral tissues and packaging it for the return journey. This reverse transport process is why high levels of the reverse transport lipoprotein are protective. It is actively removing cholesterol from places where it causes damage. The modern understanding of cholesterol risk goes far beyond simple total cholesterol numbers. What matters more is the size, density, and oxidation state of the particles carrying the cholesterol. A diet high in refined carbohydrates and sugar triggers the liver to produce more small, dense lipoprotein particles, which is why dietary patterns that spike blood sugar repeatedly are linked to cardiovascular risk, even without raising total cholesterol. Chronic low-grade inflammation makes cholesterol-related damage far worse. Inflammatory molecules that circulate in the blood make the artery wall more permeable, make lipoprotein particles more likely to be oxidized, and make plaques more likely to rupture. Cholesterol is not the villain it was simplified into. It is a molecule of extraordinary importance that your body makes, transports, uses, and recycles in a precisely coordinated system. The problem is not cholesterol itself. The problem is when the wrong particles, in the wrong concentrations, under the wrong conditions of oxidation and inflammation, get into artery walls and trigger a cascade that the body cannot reverse. Understanding this system, the particles, the oxidation, the inflammation, the endothelium, is what allows you to make choices that genuinely change the biology happening inside your arteries right now. Every heartbeat right now is pushing blood past arteries that contain cholesterol particles, endothelial cells, and all the molecular machinery we just explored. And the balance of those systems determines your cardiovascular future.